Hi, guys. Welcome to our screencast on genetics. This week, we're going to start talking about genetics, which I know a lot of you talked about a lot in middle school. So I'm going to move quickly over some things and focus on what I think you need to know as new. So I'm starting by looking at this Schmoop article. I did assign this to you guys this week. Okay. I did assign this one to you this week as a reading. So I just wanted to show it to you. It's a little bit long, but you can always kind of play around on here and see what's what. So let's get started with our notes. Okay, so we could also refer to genetics as patterns of heredity because we're talking about things that have been inherited from generations prior. And we always start by talking about Gregor Mendel, who lived in the 1800s. Interesting guy. He's considered the father of genetics because he is essentially the person who figured out how traits are passed down from parents to offspring. There's a lot in the reading about Gregor Mendel, so we're not going to go crazy. Interesting that he was a monk. He was also a statistician, meaning he studied statistics. And genetics, the way that we look at it right now, is basically statistics. He used garden peas, which he's become famous for, in order to observe how traits are passed on from parent to offspring. He looked at 7 characteristics. And this is a good time for us to talk about characteristic versus trait. A characteristic is something that can be inherited. When we think about peas and plants, we often think about flower colors. So the color of a flower is a heritable feature. That's a characteristic. Each characteristic occurs in 2 different traits. So the trait, right, would be the actual color. So yellow versus purple or white versus purple. You'll always have 2 different traits to look at when you're thinking about genetics. Sometimes more, but minimum 2. These are the characteristics that Mendel studied. The type of seeds, the color of the seeds, the color of the flower, the type of pod, the color of the pod, whether they were short or tall, and then axial or terminal flowers. And what that means is, are the flowers at the end of a stem, terminal, or are they on the sides? That's axial. So what he did was collect seeds from the pea plants, right? The peas that you eat are essentially the seeds of the plant. And he recorded all of the traits. Then he planted seeds the following year to see what would happen. His observations were that purple flowering plants grew from most of the seeds that came from purple flowers. However, white flowers also grew from those plants. The other thing was tall plants produce mostly tall plants, but some dwarf plants or short plants as well. And this is where he was observing recessive traits coming through based on which alleles or genes were passed down from the parents. I'm sure these are familiar to you, right? Punnett squares. We'll be doing a little bit of work on these this week. Okay, now this is what I want you to look at, right? The ratios, because when we do our Punnett squares, no matter what kind, what we always end up looking at is the ratio. The ratios are probabilities, meaning not what you're definitely going to get, but it's possible for what you're going to get every time a plant reproduces. Okay, so his hypothesis, which turned out to be supported by data, was that something in the plant controlled which characteristics, traits were observed in offspring. Mendel called them factors. Now we call them alleles. And alleles are those 2 corresponding genes, essentially, right? Dominant and recessive. He hypothesized that each trait was inherited by means of a separate factor or a separate gene, and that is true. And he hypothesized that a pair of factors or a pair of alleles controls each trait. And again, that's true, right? One from mom, one from dad. This brings us to the idea of recessive and dominant, which I'm assuming you guys are all familiar with. Dominant traits is a trait that will prevent another trait from being seen or from coming through. It will mask the other traits. So, for example, let's go back for a second. Right? These are all of our dominant traits. These are our recessive traits. So, what that means is this trait will mask this trait unless you have two copies of a recessive. And then recessive traits will appear with two copies of a recessive factor or a recessive allele. It has no effect on an organism's appearance or traits when paired with a dominant allele. So you can see, right, true breeding means homozygous. So homozygous meaning two copies of the dominant allele, two copies of the recessive allele. When you have this cross, you will only see the dominant trait. It's not until the F2 generation, so when you breed these guys, that you'll see this recessive trait come out. Okay, this brings us to two laws. We have two laws that we talk about here, the law of segregation and the law of independent assortment. What this means is paired factors, paired alleles, right? Let's say dominant and recessive, separate during the formation of the gametes because you're going from diploid to haploid when you're talking about the formation of gametes. So think back to meiosis last week. Then, when the gametes combine, you come back to two alleles for each characteristic. So here's the definition for the law of segregation. And the second one, the law of independent assortment, also relates to meiosis, meaning factors separate independently of one another during formation of the gametes. So when we talked about, not all of mom's genes and not all of dad's genes lining up on the same side of the cell before those cells are split, that's what this law is referring to. If you have dominant traits, they don't necessarily appear together. If you have recessive traits, they don't necessarily appear together. Alleles or factors for individual characteristics are not connected, meaning just because you have blonde hair doesn't mean you can't have brown eyes. They're not, they don't necessarily go together. And again, this is independent assortment in meiosis. We're just thinking about it kind of in a different way here in this unit. Okay, and then finally, alleles, right? Sometimes, at least in this notes presentation and the way that Mendel referred to it, they're called factors. These are genes when we talk about alleles. The alternate form of a gene is called an allele. So, for instance, this is an allele and this is an allele. You would get one from mom and one from dad. And we'll talk a lot more about that as we get into doing Punnett squares and evaluating crosses, genetic crosses. All right. And again, I know I've said this a million times, I'll say it again. During meiosis, gametes get one chromosome from each homologous pair of chromosomes. So when they are combined during fertilization, you end up with one from mom, one from dad that will actually make the new organism. Okay. These are the possible gametes from these particular alleles. This is a little more complex because this is what we would call a dihybrid cross, and we will get to that. So don't worry about that for now. And then one of the things that I'll be asking you to do this week is to start actually working on some of those monohybrid crosses or the simple Punnett squares. I'm pretty sure you guys have done this already, but we can certainly come back together and talk about that another time. Okay. Real quick, A couple of important definitions: genotype and phenotype. Genotype is the genetic makeup. It's the actual alleles that an organism inherits from its parents. So it's not necessarily what you see, it's what's in the DNA. And then the phenotype is what you see, or the expression of the trait. So I always think P for phenotype, sorry, P for physical, because it's generally a physical manifestation of those genes, of your genotype. Just another thing that I'm sure is a review: homozygous versus heterozygous. Homo means the same. So when both alleles of a pair are alike, that is when we say homozygous. So here, right? Homozygous recessive, homozygous dominant. You can also refer to these as true breeding. Some resources will refer to these as purebred, but all of those things mean homozygous recessive, homozygous dominant. And then heterozygous, hetero is different, right? So when you have two different alleles in a pair, so one dominant, one recessive, is heterozygous. And that will show itself in a phenotype differently depending on what the trait is. But most of the time, what this will give you is a dominant phenotype. So we would refer to this as heterozygous dominant. You don't even have to say the dominant, because heterozygous kind of implies that, but that's what you would see in that phenotype. Okay, I'm going to leave off here. I'm going to let you guys work on some Punnett squares. This notes presentation is posted, and there are a lot of Punnett squares that kind of happen as animations. So these would definitely be good for you to take a look at on your own and then do a little bit of practice. You have a lot of practice to do this week as well. All right, that's it for now, guys. I will be back to talk about dihybrid crosses and some of the other kind of exceptions to the rule. And that will be coming soon. Thanks. Bye.